question time.
Thank very nice presentation and also very good attempt. You know that we can hear most of the time bees honey adulteration is very popular. What are those adulterants?
Yes, thank you, madam. Some adulterants are water, mainly use water, but some companies, like people, small scale, these people used added some chemical composition also. They are adding this chemical component. They are objective to enhance the shelf life, this honeybee.
Okay, considering that bees feeds like those things.
Sorry, madam, can you repeat the question?
Considering this type of feeds, I mean sugar, like those kind of things, I mean. Okay, never mind.
Can I ask a question? Or,
yes, Doctor.
Okay, Ms. Bandara, thank you for the very nice presentation. So, can you actually explain a little bit why would you see a different behavior or a cleaning behavior, or why these drone bee populations are different? So, what would be the reason? So, you mentioned about the protein. Will that be the only reason for this cleaning behavior differences and things?
No, madam. After this behavioral observation, I observe during this research period, because these are after the increment of drone bee population, they like to swarm in. Therefore, their population increment, and they need to replace another colony.
Because of the okay. So what's the difference? The cleaning behavior.
Cleaning behavior, like the honeybees clean their bottom board and their brood boards like structure. They are themselves. They want to remove their debris on the box.
But you saw a difference. Sorry. Yeah. So you saw difference between treatments, right? So the cleaning behavior is also different between treatments, right?
No,
yes, T5 and T6.
No, madam. Higher level cleaning behavior observe these treatments, but another treatments also have some cleaning behaviors. But highest colonies, I observe these treatments.
Okay, so there should be something. There should be a reason for that, right? So maybe for future. So another small question. So did you test? Did you do any tasting of these honey resulted from these feeding?
No, like, madam.
Because there could be some differences in the taste as when you feed something to the honey other than flower of the pollens, right?
No, madam, I can't evaluate honey quality.
Yeah. Okay. Yeah. But better to do it in future, right? Do you think, right? Yeah. Okay. Thank you.
Thank you, Ms. Bandara. It was a nice presentation. I also had the same question as with Dr. Upamali. I think you should measure the quality and also, the taste and everything. And also, what do you think now? You have used substances like ingredients like soya bean and things like that. So there will be some, individuals who are allergic to soya products like that. So did you consider that when you are designing the experiment? And also I think this is also a suggestion for you in future. one thing is you went very fast in your in what you call this methodology, and we could not follow the treatments. So I would like to see your results slide again, please. Okay. So this is diet consumption weight, and you have seen that the consumption weight is higher in T1, T6. In this parameter, were you able to measure the these were introduced as supplementary diet, and you are measuring the diet consumption, and that is based on the weight of the diet that you provided. And how did you measure the natural weight loss? Not due to the feeding of bees, but there may be some natural weight loss, right, in your feed. So how did you tackle that?
Yes, madam. During this study, I measure this reducing after we consuming weight only, but the natural weight loss is not considered.
Okay. Can we go back to your treatments, please?
In the results, madam.
Yeah, this is the result. Yeah, this is what I actually wanted to ask you. Can we go back to the treatment, the treatment slide in the methodology? Okay. So your what you have observed was that treatment, if I remember correctly, 1, 5, and 6, right? What were the treatments that you are recommending as good supplementary diets?
I recommended treatment 6 and 1, madam.
6 and 1. So soybean flour 50 grams, sugar powder, lime syrup. Okay. Right. Okay. Okay. Right. Okay. Thank you.
Right. Thank you very much. So thank you very much, Ms. Bandara, once again for your nice presentation. With that, let's move to the next presentation. As there's a connection issue with the next presenter, let me now invite Ms. DMD Bhagya, index number 196021. Development of alginate-based antioxidant packaging films incorporated with Kanda, Makaranga peltata, leaf extract. Yes, Ms. Bandara. Sorry, Ms. Bhagya, you can start now.
Yes, sir.
Can you share your presentation, please? Ms. Bhagya?
Yes, sir. Still, I'm sharing my presentation.
Okay.
Sir, please give me a second.
Okay. Ms. Nayani?
Yes.
Miss Naini? Sorry, Miss Bhagya. Can you hear me? Please unmute your mic. Right. Now we can see you. Please unmute your mic.
Okay, sir.
Right. Now you can continue. We can hear you.
Good morning, everyone, and welcome to my presentation. I am Dilni Bhagya, index number 1960-21. To start, let's look at this slide. What comes to your mind when you look at these pictures? Yes, you are right. These are food packages. Among these, plastic food packages play a major role in food industry. But I think you all know that traditional plastic packaging has several issues like non-biodegradability, waste management challenges, marine pollution, impact on wildlife, recycling limitations, and health issues. To overcome these, we can introduce sustainable packaging. Based on that, here is my research topic. Development of alternate waste antioxidant packaging films
Miss Bhagya, could you please share your presentation again?
She lost the
Okay,
connectivity
sir.
probably.
Yeah.
Okay.
Yes. Please share your presentation now. Can you hear me, Miss Bhagya?
Yes, sir, I can hear you.
Yeah. Please share your presentation. And start at the point you stop your presentation.
Sir, this slide does not.
Yes, please. We can hear you. Please continue.
Okay.
Miss Bhagya, yes, you can talk now. You can present now.
Ms. Bhagya, can you hear us?
Miss Bhagya, can you hear me? I think there could be a connection problem. So with that problem, shall we move to the next presentation? Let me invite the next presenter. Miss Bhagya can continue later. Let me invite Miss Buddhishani, Miss KAERM Buddhishani, index number 196023. Effect of chemical treatments on germination of Withania somnifera seeds under in vitro and in vivo and in vitro conditions. Miss Buddhishani?
Yes, sir.
Yes, we can hear you. Please share your presentation. Ms. Buddhishan, please go to the present. Yeah, now we can see your slides. You can start.
Okay, sir. Good morning, everyone. Today I'm here to share the findings of my research. I am Maheshika Buddhishani under index number 196023. My research focused on effect of chemical treatments of germination of Withania somnifera seeds under in vivo and in vitro conditions. Withania somnifera, most commonly known as a ashwagandha. It's a perennial shrub belonging to the family Solanaceae. This plant is promising medicinal plant with a long history used in Ayurvedic medicine. Ashwagandha is a natural remedy because it contains certain properties association with stress management, inflammation reduction, antioxidant activities, and combat the effect of free radicals in the body. Recent studies suggest it has some properties to cure certain diseases, such as musculoskeletal conditions, bronchitis, neurological disorders. When considering the cultivation of this valuable plant, it's mainly propagated by seeds. But germination of ashwagandha erratic and limited due to the hard seed coat create the physical dormancy. As a solution for the physical dormancy breaking, there are two ways to break the physical dormancy. First one is temperature shock, and second one is providing of the pre-germination chemical treatments. In this study, mainly focused on the pre-germination chemicals treatments. In this slide, you can see what are the objectives of this research. Now look at the materials and methodology. This research conducted in Faculty of Agriculture and Plantation Management, Wyoming University of Sri Lanka. Seeds were collected from the Bandaranaike Memorial Ayurvedic Research Institute, Maharagama, and the Ayurvedic Research Herbal Garden, Ambevela. Optimization of ashwagandha seed germination tested in two different environmental settings. First one is in vivo condition and second one is in vitro conditions. Now look at the optimization of in vivo seed germination. There are three experiments were conducted with three different pre-germination chemical treatments. First one is gibberellic acid, second one is potassium nitrate, third one is sodium nitrate. In this chart, you can see what are the different levels used in each experiment. The seed pre-soaked in different levels of each pre-germination chemical treatments for 24 hours and the seed soaked in the water for 24 hours as a control. In seed sowing, the soil mixture prepared using topsoil, coir dust, sand, and compost. After that, seeds pre-treated with the fungicide solution before sowing. After seeds treatments, 2 seeds were placed in the plug tray cell. For data accuracy, each experiment repeated 3 times. Now look at the optimization of in vitro seed germination. Only one experiment conducted in vivo condition using gibberellic acid. First, sterilization was practiced using multi-step protocol. First, thoroughly wash with soap and water. After that, sterilize using fungicide solution, 70% ethanol and 10% Clorox with the 2020, and finally thoroughly wash with the distilled water. After that, seeds are treated with gibberellic acid, the same levels used in the in vivo, and for 24 hours, and further sterilization was practiced. Media preparation, used half ms media supplemented with BAP, 2,4-D, sucrose, and agar. Pre-treated seeds were cultured in the jam bottles and maintained the specific conditions. In data recording, germination counts were recorded weekly till 35 days, both conditions, and after that, the germination percentage was calculated. Should height, diameter, number of leaves were measured till 35 days weekly in vivo. Experimental design was complete randomized design. A statistical analysis conducted using RStudio statistical software, ANOVA used for the data analysis, and mean separation done using Fisher's LSD test. Now look at the results and discussion under in vivo seed germination. First time talk about effect of gibberellic acid. Ashwagandha seed germination significantly impacted by tested levels of gibberellic acid on germination. All the tested levels of gibberellic acid enhance the germination percentage of the ashwagandha seed than the control. Control record the least germination percentage. It was 2.6%. Now look at the effect of potassium nitrate. there was a significant impacted by different levels of, there was no significant impacted by the different levels of potassium nitrate on the germination percentage, as well as there was no significant impact on the different levels of potassium nitrate on the growth parameters. Third one is effect of sodium nitrate. There was no significant impact on tested levels of sodium nitrate on germination percentage, but there was a significant impact by the tested levels on growth parameters when consider the number of leaves. The highest mean number of leaves recorded in the 0.8 grams per liter, the lowest mean number of leaves recorded in the control. When consider the shoot height, the highest mean shoot height recorded in the 0.4 grams per liter and the lowest shoot height recorded in the control. When consider the shoot diameter, the highest mean shoot diameter recorded in the 0.8 grams per liter and the lowest shoot diameter record in the control. When consider the in vitro seed germination, the highest mean germination percentage recorded in the 2 grams per liter. The germination percentage was 0 at the levels of control 1 and the 2.5 grams per liter levels. So, gibberellic acid shows a significant positive impact in ashwagandha seed germination under both in vivo and in vitro conditions. The level of 2 grams per liter results in highest mean germination percentage under in vivo and in vitro conditions. The level of 0.8 grams per liter of sodium nitrate was beneficial for the seedling development in vivo. As a conclusion, gibberellic acid optimized the ashwagandha seed germination in vivo and in vitro conditions. Sodium nitrate was beneficial for seedling development in vivo. This is the time to, I would like to acknowledge the people who helped in this research period. This is the reference I followed. This is the end of my presentation. Thank you.
Thank you very much, Ms. Buddhishani, for your nice presentation. And now it's question time.
Thank you, Ms. Buddhishani, for the interesting, very nice presentation. So why didn't you talk about potassium nitrate at the end? So can you go back to the potassium nitrate results slide, please? Yeah. So don't you see any difference in the shoot height, number of leaves? You said there's no impact on any of these by potassium nitrate, right? But
Yes,
there are
madam.
some differences.
It's most of the levels are not difference in the levels. It's not some impacted on the germination or the number of growth parameters.
Okay, so, but there are some differences in shoot height and number of leaves, right?
Yes.
You can see it, right?
Yes.
In the results? Yeah, okay. So my question is, where do usually these ashwagandha grow? What climatic conditions in natural environment?
It's mostly growth in dry zones. It's mainly growth in India, the dry zones of the India. So it's mainly grow in dry zones with well-drained soil.
Good. Thank you.
Any more questions from the judging panel? Right, if not, we can move to the next presentation. Thank you very much, Ms. Buddhishani. Now let me invite Ms. DMS Dasanayaka, index number 196028. The title is Introducing Stingless Bees, Tetragonula erydipenis for Improving Bitter Gourd, Momodia coordination in Ms. Dasanayaka. Ms. Dasanayaka, can you hear me?
Yes, sir, I can hear you.
Can you please share your presentation?
Yes, yes, sir. But I logged from lab, but it did not allow me to unmute the mic.
Right. Okay. Then please contact Ms. Subhashini. Yeah. Yeah. Please contact Ms. Subhashini. Then you can join us later, right? So with that issue, let me move to the next presentation. Miss EGSS De Silva, can you hear me? Miss De Silva? Miss De Silva, can you hear me?
Yes, sir.
All right. Could you please share your presentation?
Okay, sir.
Right. So index number 196030, Miss EGSS De Silva. Her title is Developing Tania Xanthosoma sagitifolium starch-based biodegradable films incorporated with soursop, Annona muricata leaf extract for active packaging applications. As we frequently face connection problems,
So you are
Ms. De Silva, can you
sorry,
hear us?
sorry,
Sir,
Miss
can you-
Silva.
only for the last bit, sir. Only for the last bit.
Yes, Miss Silva, can you hear me?
Yes, sir. Sir, can you wait a little bit?
Sorry,
Can you wait a little bit?
you want me to wait a little bit?
Yes, sir.
You need more time, right? Okay. You can share your presentation leisurely. Number 196030. Is there any connection issue?
Sir, my screen has stopped. So,
All right. So with that issue, shall we move to the next presenter? Miss Dulmini, to save the time, shall we move to the next presenter? Miss Dulmini, can you hear me? Miss Dulmini? Miss Subhashini, can you contact Miss Dulmini?
she's there, sir, and we already make her a co-host. Miss Dulmini, can you hear us?
Miss Dulmini, please unmute your mic. I don't know whether you mute your mic.
Yes, sir. In previous time, I cannot unmute one. That's why.
Right. Okay, Miss Dulmini, index number 196041. Effect of naphthalene acetic acid and light on in vitro shoot multiplication of Dioscorea alata. Yes, Miss Dulmini, you can start now.
Sir, can you see my slide?
Yes, we can see your slides and we can hear you. No issue.
Good morning, everyone. I'm Isanka Dulmini, index number 196041. Today, I'm here to share the finding of my research on effect of naphthalene acetic acid and light on in vitro multiplication of Dioscorea alata. Do you know these name of these yam varieties? Angiliala, Hingurala, Rajala? I think most of your answer is no. Just like me before starting this research. These are few examples for the Dioscorea alata. Dioscorea alata in worldwide there are many names for this, like great yam, white yam, water yam. In Sinhala we call as Mahavellala. So it is a tropical tuberous crop and it having many more importances. Most of the African and Asian people use this as their staple food source due to nutritional and medicinal value. So it having drought tolerance and less pest and disease problems, and ultimately it gives high yield. So they are, as well as they're having some industrial uses such as flour production, coloring production, but they're having a limitation in commercial production in yam cultivation due to conventional propagation method. Here you can see the tuber cuttings that use as a common conventional propagation methods, but when during the tuber cutting, it can cause for the disease transmission. And this is a seasonal crop. We cultivate in Yala season and harvest in Maha season. Therefore, we can obtain year-round planting material. So, as a potential solution for that, the in vitro multiplication can be introduced. So, based on that, my research was the optimization of the medium components and culture conditions for in vitro multiplication of Dioscorea alata. The specific objectives were the study the effect of NAA concentration, light on in vitro shooting and rotting, study the effect of light on tuber initiation. Here are the location and duration for my research. First, under the methodology, I have selected nodal segments from in vitro stock plantlets as explant. Then I have prepared culture media. For preparation of the culture media, first I prepared half strength MS media by adding 2% of the sucrose and 4 concentration level of the NAA as treatment level. Then after adjusting the pH, the added agar to final volume of the media, thereafter microwave and dispense to culture bottle and autoclave at these conditions. So under the laminar flow, the explants were cultured on culture tubes and the cultures were maintained under these culture conditions. For the testing of the effect of light on multiplication, selected medium based on the Na concentration used for the presence of the light and absence of the light. Here are the parameters I have used for the data collection. A completely randomized design used as an experimental design. The RSD used as statistical software tool. The data analysis was done by using ANOVA and mean separation done by using HST test. Now, we are in the important part of my research presentation. It is the results and discussion. First, I will begin by discussing the effect of NAA on in vitro shooting. Let me give some example in the explant response percentage. According to the graph, you can see the addition of the NAA significantly increased the explant response percentage for the shooting. Between them also, 0.5 and 1 milligram per liter NAA gave higher response percentage. When come to the days to bud break, in here you can see in control treatment, it required longest time duration to bud break. Addition of the less concentration of the NAA also caused some delay in the bud breaking rather than other concentration. But 1 milligram per liter gave quick bud break. When consider about NA concentration versus plant height, here also the addition of the NAA has increased the plant height, but there is no any statistical difference in plant height according to the applied or tested NAA concentration. Now, in here, similar to other previous one also, the addition of the NAA increased the number of shoots per explant and number of leaves per plant. Now we will discuss about the effect of NAA on in vitro rooting. As usual, go for the explant response. In here you can see the addition of the NAA, all tested NAA concentration gave the higher response percentage, it is 100%. And the control treatment gave only 94% response rate for the rooting. And similar to days to bud initiation, for the root initiation as well, the control treatment required a longest time duration to root initiation. When consider about number of roots per plant, in here, The 1 milligram per liter Na concentration gave higher number of roots compared with the other added concentration and the control. So now we discuss about the effect of light on in vitro shooting and rooting. In this bar here you can see in yellow color the number of leaves, shoots and roots are increased rather than the dark condition in light condition. But come to the plant height, there is no any statistical significance during the light condition and the dark condition. In this picture also you can see the similarity of the plant height in both light condition and the dark condition. So when discuss about the effect of light on in vitro tube initiation, the signs of tube initiation could be observed under the dark conditions from aerial tubers. Now, according to the results, we will come to the discussion part. According to the result, it is clear that NAA application significantly enhance the proliferation of buds, leaf, shoot, and root production efficiency. When compared with the previous findings, it mentioned that there were no any bud initiation or development in the media without any growth regulators. But in this experiment, we can obtain in vitro plantlets with very good growth in culture media devoid of growth regulators. As well as some previous literature said that the addition of the IAA or NAA improved the shoot growth. So in my experiment, it completely supported for the shoot growth in addition of the NAA. So let's come to the conclusion. The optimal NA concentration for the in vitro shoot and root multiplication was 1 milligram per liter. The shoot and root multiplication increased with the light condition, but the complete darkness negatively affected for the growth and development of plantlets, but it tends to initiate in vitro tubers. So I want to acknowledge the YMB University of Sri Lanka for providing financial assistance and all the academic and non-academic staff members of Department of Horticulture and Landscape Gardening. And here are the references. And finally, thank you everyone.
Thank you very much, Ms. Dulmini, for your nice presentation. And now it's the question time.
Thank you, Dulmini, Ms. Dulmini, for the nice presentation. So how long do we have to wait to plant them in the field? Do you have any idea how long will it take to do the hardening and then to get them in the field?
Yes, madam. In this research, I wanted to plant a plant in vitro conditions, but it is need to test in vivo conditions. But I think another research is needed to find out the other conditions for the hardening or acclimatization of the in vivo condition.
So to do this trial, actually, did you use the tubers from ground or just the aerial tubers? Sorry, I didn't catch that part, I think.
And in vivo conditions, from the aerial tubers, aerial roots, the tubers were initiated. Here you can see in this slide also, the picture shows the tubers initiated.
No, I mean for the trial, what material did you use for the initial trial?
It's in vitro stock plantlets.
Plant.
Yes.
Okay, that's fine. So you didn't get the question. That's fine. So that's only for the purple color Rajala variety, right?
Yeah, I test for the Rajala variety.
Rajala. Yeah, that's good because yeah, that's really hard to find the purple color planting material when we are really going to the planting the purple variety in the field. So it is good that if we can actually have tissue cultured plants and then we can see whether they are going to perform better in future as well, right? So thank you for the nice presentation.
Thank you.
Ms. Dulmini, thank you. It was a very nice presentation. I guess Rajala is having a lot of pigments. Did you find any difficulty in this in vitro culturing because of these pigments in your explants?
No, yes, some browning effect was observed, but it was not much problem to initiate the shoots or the tubers.
Okay.
So it can be avoidable.
Okay, right, thanks.
Any more questions?
Miss Dulmini, very nice presentation.
Miss Anoma, I think there's a connection problem. We can't hear you properly. Miss Anoma?
Yes. Can you
Yes, now we can hear you.
Miss Dulmini, very good job and also presentation. Do you know the harvesting time of the Rajal? I mean,
Excuse me,
period.
madam, I
How long
cannot
we
hear.
can keep? We have.
Miss
Hello,
Anoma,
madam.
could you please repeat the question? It's not clear. Are you asking about the harvesting time of Rajala?
Yes, Rajala, yes.
Yeah. Yes, Dulmini. What is the harvesting time?
Yes, normally we cultivate in March to April months, and we harvest this in December to January period.
Okay, thank you.
Thank you very much. Thank you very much, Ms. Dulmini, for your presentation. With that, we can move to the next presenter, Ms. WSW Fernando. Can you hear me?
Yes, sir, I can hear you.
Yes, thank you. Index number 196048. Miss WSW Fernando. Title of her research is Split Crown Technique for Rapid Propagation of Pineapple, Ananas commosus, variety Mauritius in Sri Lanka. Yes, Ms. Fernando, you can share your presentation. We can see your slides. Now you can start.
Okay, sir. Good morning, all. I'm Subhita Fernando. First of all, I would like to ask you a question. Do you know about the prince who wears a crown in the fruit world? Can you guess it? Well, as we all know, that's pineapple. He wears a crown. So today I'm going to tell you a hidden secret about the pineapple. The pineapple plant Ananas commosus belongs to family Bromeliaceae. It's a tropical fruit-bearing plant with spiky sword-shaped leaves, and it has a unique fruit structure. There are so many pineapple varieties in the world. In Sri Lankan scenario, pineapples hold significant importance in agriculture and economy. But the problem is mass-scale production of quality pineapple planting materials have some constraints because conventional methods of propagation have slow rate of multiplication and tissue cultural propagation has some mutations. Therefore, as a solution for that, the pineapple crown was suggested to test whether it can be produced more suckers when applying some treatments. So, in sometimes, do you excited when I say that? Because it's really hard to get suckers by using crowns. So, why did I use crown for that? Because the pineapple crowns are normally disposable item of pineapple and it can be collected easily cost effectively. The most important thing is the pineapple has been proven to be free of viruses in a laboratory test. Therefore, due to the difficulty in obtaining planting materials using crown, the split crown technique was applied for that. The split crown technique has been reported is an efficient method to obtain suckers when applying to the crown. Therefore, I thought of doing a research by using split crown technique for rapid propagation of pineapple using Mauritius variety in Sri Lanka. In the objectives, the general objective was enhancing mass-scale planting material production of pineapple crowns And specific objectives were selection of the most effective crown preparation method and to determination of the most effective indole butyric acid hormone concentration. The research was conducted at Fruit Research and Development Institute, Kananvila Horana, and all the experiments were conducted in a protected house. When talking about the method, this research was conducted in experiment 2. In the first experiment, initially, the pineapple crowns were collected using Mauritius variety due to its high productivity, and then topsoil and compost were mixed in 1:1 ratio for preparing growing media. Then, all the selected crowns were prepared according to 4 treatments. You can see it in the graph. In the first and second treatment, removal of apical bud and crowns split into 4 sections and 2 sections respectively. And in the third treatment, the crowns were subjected to removing apical bud and without splitting. In the fourth treatment was considered as the control one. Then, after the preparation of crowns, all the crowns were treated with fungicide solution for 5 minutes. And after 1 week of planting, a foliar fertilizer was applied for all the crowns. Then, at the end of the experiment 1, the most effective base treatment was selected to conduct the experiment 2. In the experiment 2, initially material collection and preparation of growing media were done as in experiment 1. Then all the collected crowns were prepared by removing apical bud and split into 2 sections. And then all the prepared crowns were treated with fungicide solution. After that, indole butyric acid hormone was applied in 3 concentrations. You can see it in the table. In the process of data recording, after 1 month of planting, the number of newly emerged suckers were counted in both experiments, and the horticultural characteristics of suckers were evaluated.